Good afternoon, dear participants. On behalf of Annakkili Research Institute, Chennai and e-teaching Tamil, I am very glad to welcome you all to the 16th session of ARI Bioscience webinar series. I am very much proud and happy to introduce today's special guest, Dr. AJAY G. NAMDEO, Associate Professor and Head, Department of Pharmacognosy, Poona College of Pharmacy, Bharati, Vidyapeeth, Pune. Dr. AJAY NAMDEO did his Masters in Pharmacognosy from Dr. Harit Singh Gore University, Sagar, and PhD on Bioreactor Management from Devi Ahilya University, Indore. He has an experience of industrial research in collaboration with Baba Atomic Research Center, Mumbai, and he has done MBA Marketing and MSc in Total Quality Management. He completed the DPT's overseas postdoctoral associateship under the Professor Dr. R. Verpurte in Leiden University, the Netherlands, and worked on plant metabolomics. Dr. NAMDEO has completed 4 government-funded research projects worth rupees 70 lakhs from DBT, AICTE, UGC, and BARNS. He has guided several PhD and postgraduate students. He has published 50 research papers in national and international journals, and presented 80 research papers in international and national conferences, and delivered more than 75 guest lectures. Dr. NAMDEO has about 1300 citation index. He has authored a reference book on medicinal plant biotechnology and written 3 book chapters and contributed in monograph entitled American Herbal Pharmacognosy. He has visited several nations for various academic and professional activities. He is life member of many professional societies. With this short introduction, I would like to call Dr. AJAY G. NAMDEO sir to present his talk on herbal drug technology. Sir, the floor is yours, sir. You can continue.
Okay. Okay. Thank you, Parimal. At the onset, I want to thank the organizers of Annakkili Amma Research Institute, ARI, and Biosciences for inviting me to deliver the talk on herbal drug technology. In fact, in the present era of COVID-19, all of us are suffering from a very severe situation of continuous contamination, infections, death, and the entire lives, not only in Poona or in Maharashtra or in India, the entire globe is suffering from this particular virus. And it has been long now for last 6 to 7 months, whereby it is increasing day by day. The number of cases, if you see the screen, this is yesterday's count. So they are gradually or drastically increasing. Now, in this view, when Corona is there, earlier it was a concept of herbal drugs or medicinal plants. We used to use this, but in this particular scenario, the herbal drugs come up with very prominently, whereby the government of India, the Ministry of Ayush, they have suggested that the COVID virus can be overcome, or the attack of COVID virus can be met by the how we can increase our immune system. So they have suggested some one should take Chyawanprash, daily Chyawanprash. Then they have suggested herbal tea or kada. This consists of tulsi, dalchini, kali miri or black pepper, then dry ginger or salt, and murakka. And also they have suggested that one should drink golden milk, or the milk which is having turmeric or haldi. This we used to in our day-to-day life. We also use these spices in different forms for centuries together. However, when the present COVID scenario is unable to overcome or cured by modern medicines or modern concepts of medicines, the people are targeted towards the natural drugs or natural products. Now, if we start our session, we say that plants, they feed us, they cure us, they are help to dress us, they please us with the color and scent of the flowers. They can kill us like cyanide and strychnine are there, or they can kill us very slowly like opium or nicotine. So with this particular background, we understand that yes, plants are having certain activity which our ancestors have been using. Can we use these plants or plant products to cure diseases or corona-like diseases, and can we exploit these plants. Now, if you see the background of our traditional system, that Ayurveda and Yoga, these are the two precious gifts of India to the entire world. Charak Samhita and Sushruta Samhita, they are the treasures of literatures which suggest the indigenous medicines. Now, indigenous medicines are those which are described in old texts or Vedas. And it is also followed by Greeks and Romans who extensively wrote on the medicinal plants for healing or different activities. There are almost 250,000 plant species which are there, and which are being used by human beings. Now, if you see the plant can be used as medicine, they can be used as spices, they can be used as food material, but some of the plants, they intermingled, like Vidania. Vidania can be used as food, as spices, or as medicine, like Karela or bitter gourd. Bitter gourd, mainly we use it as food material, but it can be used as a medicine, or spices like ginger or curcuma, like haldi, carom turmeric, black pepper. So many plants are there. So there's no strict boundaries. Okay, this particular plant is only used for medicine, or only as spices, or as food products. So with this particular background, if we go further, the Ayurveda says that amantram aksharam nasti nasti malainasunam ayogyaha purusho nasti yoga durlabha. What does it says is, there is no letter which doesn't have a charm, there is no root which doesn't have a medicinal property, and there is no person who is not able, but rare is the one who knows his or her proper application. So in this particular verse, it is mentioned that all plants are having one or the other medicinal properties, but we have to explore it only. And it is also mentioned that 80% of the medicines are obtained from plant. 60% of the medicines are obtained from plants, and 80% of the population is dependent on primary healthcare. Now, if we see the plant or different plant parts, there are a wide range of examples like seeds. So, Natsamica is the example of seed, Strophanthus or Isabgol or castor seeds are there, from where we can get the castor oil. The leaves like senna, neem, vasaka, tulsi, digitalis, eucalyptus. So many leaves are there which we are using in our day-to-day life. Then barks like cinchona bark, cinnamon bark, kurchi bark, or woods like quassia wood, sandalwood, red sanders. Then if you consider root like Rauwolfia, Vidania, Ipecac, jalap, so many roots are there. Then rhizomes like ginger, turmeric, podophyllum. So these are the plants or plant parts which have been used for their therapeutic or their medicinal values. Like flowers like clove, pyrethrum, artemisia or saffron. Then fruits, different entire plant like ephedra or ergot, belladonna. The entire plant, means all plant parts are used for medicinal purpose. Then dried juices like aloe, quinoa or red gum we use. Gums, these are the unorganized plant material of the plant, or exudates, we call them as exudates, and like acacia, tragacan, gatti gum or guar gum. So these are used for different purposes, maybe directly as medicines, or as excipients, or as suspending agents, or as binding agents in herbal or pharmaceutical formulations. Then comes dried extracts like gelatin, agar-agar or katechu, and some resins like asafoetida or benzoin. So this particular slide says that okay, medicinal plants are there, and different plant parts are used. So again, it is very important that we have to find out which particular plant part from the medicinal plant is useful. In some plants, all the plant parts are used. In others, we have to just select the particular plant part for its medicinal purposes. Like you can take the example of capsicum. So capsicum, only fruit is used, neither the root, flower or leaf. They are not having medicinal values. So only the fruit is used in case of capsicum. Now, if we see the metabolism, because why the plant contains activity. So by nature's gift, the plant carbohydrates And these primary metabolites are used for the growth and nutrition purpose. They are also used for they synthesize like carbohydrates, lipids, amino acids, and they are produced in large volume. It is very important to know or to understand that primary metabolites in plants are synthesized in large volume, whereas the secondary metabolites, which are derived from primary metabolites, they are having physiological activity, but they are produced by uh, plant in small quantity. So this particular slide says that okay, and it shows the cellular economy also or plant economy also. Okay, whatever is produced in large quantity is cheap. Whatever is produced in small quantity is valuable. So this particular slide uh, gives the concept of cellular economy as well or plant economy as well. Now, if we consider the metabolic pathways from where the plant utilizes carbon dioxide and produces primary metabolites and secondary metabolites, it is a very complex phenomena. And this particular phenomena involves large number of enzymes, different steps, different intermediates. And if I ask someone, "Ki, what is your age?" So it may say, he may say, or he or she may say that, okay, his age is 25 years. But is it 25 years? No. It is 25 years, 3 months, 4 days, some hours. So it is a continuous process. Similarly, in case of plants, the secondary metabolites or the primary metabolites, they are continuously synthesized and their biochemical nature is changing. You can take the example of, say, again, a green mango or keri, or we call it keri or raw mango, green mango. If it is kept in conditions or shade conditions for 4 days, 5 days, or a week or so, then same green mango is converted into yellowish and orange so that it becomes sweet. Initially it is sour, then it is becomes sweet. So what is happening inside the plant? Because now it is plucked out from the tree and it is kept in the storage. So there its biochemical processes are going on which are continuously converting some of the primary metabolites into the secondary metabolites, and these secondary metabolites we are interested in to explore to exploit their therapeutic properties. Similarly, I'll give the example of coconut. Initially, the coconut is green, and this green coconut contains coconut water, but gradually it turns to brown, which we use for puja or in prayers. And later on, this brown part is the inside of the green part or brown part becomes dried, and out of that dried material we extract the oil. So you see the constituents which is present in the same coconut. Initially, it is water, then very good pulp, or we call it as coconut milk. Then from coconut milk to coconut oil. So this continuous process, though no machinery is utilized or used there to convert this particular metabolites into the oils or into the desired products. So it is a very complex phenomena where thousands or hundreds of
Hello participant, due to small network error, our speaker has left the meeting. He will join soon, so kindly bear with us. Give us some time.
yeah, yeah, okay. Mhm. Okay, so we are, huh? Okay, okay, I'll just check. it visible now? Is it visible now? Okay, so we understand that, okay, yeah, okay, so I'll continue. So now we understand that, okay, metabolic pathways are there, which produces different compounds, initially primary metabolites, then they are converted into secondary metabolites, some are converted into secondary metabolites, other intermediates, likewise. Now, if we consider that, now we can say that plants are the chemical factories which convert some raw material into the desired products, and there are more than 1 lakh plant metabolites which are known, and almost 4,000 new compounds are added into this particular list. It is a very uh, good that the plants are producing us the secondary metabolites or desired secondary metabolites. Now, if we consider the functions of primary, secondary metabolites in the plant, so there are, it is said that they may be used as a defense mechanism, they may be used to attract the herbivores, animals or bacteria, or for pollinating purpose, and they may be used for UV protection or for nitrogen storage. Now, if we see the few slides, next 3-4 slides, these particular slides says that, okay, what is this common? Like bears, it produces aspirin, morphine, digoxin, vinblastine, vincristine, Taxol, Senna. So what is the common in these? The common thing is that these all products are derived from plants. So it is quite interesting that, okay, plants are also used for the production of medicines or therapeutical products. Now, if we see certain examples like Vinca rosea or Catharanthus roseus, it produces vincristine, vinblastine. The yield of vincristine, vinblastine in plant is very less. It is 0.0002%. So quite less. But the phytoconstituents or the vinblastine, vincristine alkaloids, they are very costly products, as we have seen the economy of plants. Another compound like digitalis, which produces digoxin, the cardioactive product. Then opium or the morphine, which is obtained from opium or opium poppy, the Papaver somniferum. It is very strong analgesic, generally used in post-operative care. Then Vasaka, which is used for it's a phytoconstituent like vesicine, which is used for your throat infection and expectorant. So it is a very good bronchodilator as well. Then another good plant of Indian origin is Rauwolfia serpentina. We call it as Sarpgandha. So it produces reserpine, the antipsychotic drug or antihypertensive compounds are there. Then Atropa belladonna is a next plant which produces again an alkaloid named atropine, which is used for the treatment of glaucoma. Then turmeric or curcuma longa, which produces curcumin. Nowadays, curcumin or curcumin formulations are widely used and researched for their anti-cancer properties. Then Taxol, obtained from paclitaxel or Taxus brevifolia, and it is used as anti-neoplastic agent. Now, recently, if you recall, in 2015, scientist Yu from China, she got a Nobel Prize in Physiology and Medicine. And for what? For Artemisia annua. It is one plant which is used in China by traditional people. They just boil it, and whenever fever is there, they used to give this particular kada or the extract or water extract of this particular plant to people, and the fever of those people are reduced and they feel better. After continuous research on this particular plant, it is observed that, okay, this particular plant produces artemether or artemisinin, the other compound which is anti-malarial. So earlier days, for anti-malarial, for the treatment of malaria, chloroquine is given. But now, after the development of artemisinin from Artemisia annua, the new compound artemisinin are given for the treatment of malaria. Now, if you see the concept of development of modern medicine versus Ayurvedic medicines or alternate medicines or traditional medicines, this is very important, because whatever drug development is there, it is in case of modern medicine, it is a reductionist approach or structural reductionism. Means what? They take the material, they go up to its chemical entity, and they try to synthesize that particular chemical entity in laboratory. Whereas in case of Ayurvedic system, it is a holism or holistic approach. The approach in case of modern medicine is which simplifies by reductionism, whereas in case of Ayurvedic systems or in traditional systems of medicine, it is a complexity of systems approach where entire system is used. Method of development in case of modern medicines is based on, it deals with the disease, whereas the alternative medicines are deals with the health and diseases. Mode of here it is preventive medicine at nascent stage, whereas in case of traditional medicines they are for preventive medicines. The diagnosis in case of modern medicines, the diagnosis is predominantly structured, whereas in case of Ayurvedic or alternative systems of medicine, it is functional. Then treatment focus is on particular organ or parts in case of medicine, because they consider everything as matter. Whereas in case of traditional systems of medicine, it is a holistic approach, and whereas the energy as a whole is treated. The mode of treatment, however, is in case of modern medicines is treated with the mechanical interventions, whereas in case of or targeted mechanical interventions, whereas in case of traditional system, it is a comprehensive approach. Then in case of modern medicine, they do not consider the mind, body and spirit as, they consider mind, body and spirit as a separate entity, whereas in case of traditional systems of medicine, it is a unified one. Then modern medicines consider human as a machine, whereas Ayurvedic system, they consider inside itself human being as a universe. Whatever is outside, that is inside the human being as well. Then the disease is due to infection or organ defect, whereas Ayurvedic systems of medicines consider that it is the system's imbalance. The mode of treatment or the primary intervention is drugs and surgery in case of modern medicines, whereas in case of Ayurvedic systems of medicine, it is diet, exercise, herbal medicines, meditation, and manipulation. Now, if you recall this particular concept, in Ayush Ministry also suggested for to overcome or fight against corona, that one should take a healthy diet, one should minimum exercise for 30 minutes. Then the herbal medicine like Kada we are taking for the immunity boosting facility. Then they also suggested for meditation. Now, the role of medicine in case of modern medicine is to fight against infection, whereas in case of traditional systems of medicine, it is to restore the harmony within the body. And the treatment purpose is just like to seek care, whereas the total health care is the concept in case of alternative systems of medicine. Now, if we understand these particular alternative systems of medicine, now we have to consider the medicinal plants, whereby we can consider this particular slide, which I obtained from Pulak Mukherjee sir's evidence-based validation of Indian traditional systems of medicine, whereby what they have suggested that the traditional systems of medicines are there. We have to first select the plant material, identify its source, then we have to identify its historical description given in traditional text, and then we have to perform primary screening as per Ayurvedic claims, and then we have to go for its phytochemical profiling, means what are the phytoconstituents present in that, and out of that we have to just find out what are the lead identification of the lead compounds which are there, or lead chemical compounds which are there, and then simultaneously we can go for high throughput screening, where it is the concept of simultaneous analysis of phytoconstituents, number of phytoconstituents with sophisticated instruments like LCMS or HPLC or NMR, or combination of these techniques. Then the Indian system of medicines, again we can go for clinical trials, herbal pharmacovigilance, regulatory affairs, herbal therapeutics, or standardization of phytoconstituents. Now, this particular slide, standardization is very important, and in our present presentation, we'll discuss these few topics in detail. Now, but however, if you consider the plant material which is obtained from nature, so they are collected either from wild or cultivated. In India, 80% of the medicinal plants which goes to industry, they are they are collected from wild. No agricultural cultivation is going on in India. However, in case of China, because traditional Chinese systems of medicine covers almost more than 70% of the world market, and it is because they are continuously concentrating on herbal drug industry, where they use agricultural practice for its cultivation, whereas we are still fighting for that. So if the wild source of raw material is there, so there is always an inconsistency, irregular supply of the material, loss of biodiversity, because if you deharvest the particular plant from particular region, then that particular region will be devoid of that those uh, bioresources. Then the whatever raw material is collected, they are generally we order or we select the vendor and they give this for unskilled or laborers. So they may not be aware of what is the age of the plant, when the plant material or medicinal plant is to be collected, whether in the morning or in the evening, because it is uh, uh, described in literature that plants varies its phytoconstituents throughout the day. Whatever phytoconstituents present in the morning is not the same what is there in the night. I'll give you some examples like opium. If opium or poppy, if it is collected in the morning, early morning, 3 o'clock or 4 o'clock in the morning, then the phytoconstituents opium or morphine concentration is very high. Whereas in case of ratrani, I don't know what is called it, that particular plant blooms in the night around 8 o'clock or from 8 o'clock, 9 o'clock till the midnight, and it gives very good fragrance. So, and the entire day that particular fragrance is not there. So that again, the time of collection is very important. Now, if we consider the cultivation, if we cultivate the plant material, then there are various plus points like we can use agricultural practices, good agricultural practices, good harvesting practices. We can select the good material, good irrigation. Then we are able to harvest it in proper time. We can take care of the weeds, and we can restrict the use of fertilizers or chemicals. So these variations are there, or by which we can control. After this comes the post-harvest technology, or good harvesting practices are there, where we have to select the material. Like in case of glycoside-containing drugs, they are to be collected and dried below 60%, 60 degrees centigrade. If it is above 60 degrees centigrade, the phytoconstituent or the glycosides present in that, or because of hydrolysis, the phytoconstituent get degraded. So we cannot get the material of desired quality. Then degradation is again a big problem. Another some processes are like age of the plant, uh, energy loss, lack of protocols. So these are some of the bottlenecks where we can they restrict us to for herbal drug research. To overcome this, the herbal drug technology is the concept. Now I'm coming to the actual presentation. The herbal drug technology is the main concept whereby we can study various uh, parameters, and this is just a bird's eye, whereby we can concentrate on consideration of crude drug or medicinal plants, their extraction, isolation, and purification, herbal cosmetics, drug discovery from plants, herbal drug regulations, phytochemical analysis using modern analytical tools, medicinal plant biotechnology, phytopharmaceuticals and markers, then aromatherapy, herb drug interaction, nutraceuticals and dietary supplements or functional foods, herbal formulations, organic cultivation or good agricultural practices, plant metabolomics. These are some Yeah. So should I continue? Okay. Okay. Yeah. 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 So yeah. So these are huh? Thank you. Disturbance. Now these are the basic inputs of herbal drug technology. Out of this, I'll be discussing in this particular presentation like standardization of uh, crude drugs or herbal material, extraction, isolation, and purification, drug discovery from plants, plant metabolomics, and medicinal plant battery. These are the 5 topics which I am going to discuss one by one. Now, herbal drug standardization. Why herbal drug standardization is important? Because the industry or the people, they want the plant material or the product, herbal formulation, whatever is there, it should be authentic, it should be pure and safe, and it should pass through quality control or quality assurance uh, platforms. Now, if we see the raw material, this is the slide which I obtained from CSIR, one publication from CMAP, and they have distributed the parameters in 5 like botanicals, where the external features marking. Hello? Yeah, yeah. Okay, okay, okay. Requisite of our product. Now, for raw material, again, this is a repetition of similar slides, whereby the plant material collection of plant part. This, as I have told you earlier, the desired plant part is very important. If we are not able to collect the desired plant part, then we cannot get the desired phytoconstituents present in the material. So, authentication is season, region, then time of collection, drying, preparation. So these are primary inputs, and this is again all the features like chromatographic profile, loss on drying, foreign matter. Foreign matter is important. One is foreign matter, another is foreign organic matter. So foreign matter is one which is maybe like other plant material or uh, dust or dirt or stones which are collected along with the drug material, or excreta of the animals or dead insects. These are foreign matter. Whereas foreign organic matter is the term which is used for the same from the same plant but undesired material. Suppose we are interested to collect ginger rhizome. So along with ginger, the stem or the root, stem or the leaf of the ginger is also coming along with that. That is the foreign organic matter in that case. The sensory evaluation of the raw material, then it's the ash value. Ash value is very important feature or phenomena which says us or which indicates the presence of inorganic material which is present. Or if we go for acid insoluble ash, then that particular term reflects the presence of silicates present in the sample. It is basically used for evaluating the root drugs, whereby the root contains some dirt or dust, and if we calculate the acid insoluble ash, then this acid insoluble ash or the value of acid insoluble ash signifies the presence of silica or dust or mud in the sample. Then extractive value is again an important phenomena, whereby we take particular, this we'll discuss in the extraction also, but hereby we take the one particular solvent, say water is the solvent, and we want to calculate the extractive value. So in that case, we have to just check the raw material and how much percentage of the phytoconstituent which are soluble in water. Then volatile oil matter. Volatile plants contain some aromatic compounds, so they can be calculated as volatile matter present in the sample. Then again the bitterness value. Which one is more bitter, bitter gourd or neem? So it is very difficult in qualitative terms. So how to quantify them? So for that again, World Health Organization has given us the parameter by comparing the taste with that of purine sulfate. We can check the bitterness of the given sample. Swelling index and foaming index are the basic property or physical properties of the sample, whereby the water is absorbed in case of swelling index, or in case of foaming index, the presence of because of the presence of saponins, the froth formation takes place. Sometimes the plant may contain radioactive contamination. So again, that is again to be standardized, or that precaution or that evaluation is to be carried out. And heavy metal ion and pesticide we have already discussed. Now, in case of process, we know that process should be authenticated. Then the size of mixing or schedule of mixing, time of mixing, temperature, humidity. Then in process, quality control like pH, viscosity. So these are various parameters depending on the which type of formulation we are going to prepare. And overall exercise on the entire distillation process is that the patient or the doctors or the society wants batch to batch consistency or uniformity of the product. The entire process should be reproducible and repeatable. So these are the prerequisites of this distillation process. Now, distillation of herbal drug substances and herbal extracts are the two things whereby we define the botanical source, or which part is used, and in which stage, whether the plant material is fresh or dried or exudate. Then characters like organoleptic, macroscopic, microscopic, then identification on the basis of various chromatographic or chemical reactions, then different tests which we have seen in previous slide, like foreign matter, acid insoluble ash, total ash. These parameters are to be evaluated. Then contaminants. What are the contaminants? Whether constituents are presented in the sample or in the formulation or not. Then in the same plant material, the geographical variation, maybe there are seasonal variation, maybe that. I'll give you this example in plant metabolomics. Then physical or chemical contamination, and we want to assess the quality, purity, and stability of stability studies. Now this is the Ishikawa cause and effect of diagram for variation in herbal drugs. So if we particularly prepare this particular slide or this type of study, then what are the factors which may affect the our sample, like man, machine, stability, raw material, method. So these all influence on the process or on the product. For example, we take raw material. So whether the raw material is the desired raw material, who has collected the raw material, what is the time of collection, from place of collection, method of collection, identification of the raw material, so whether it contains certain substitute or not. So this is again a part of collection. Then substitution. Sometimes the entire raw material is substituted by the other cheap product. Like in case of saffron, normally saffron is a very costly product. So saffron is uh, contaminated by so many artificial things, which is which are the adulterants on them. Again, in the case of say example of honey, so honey is again a raw material which is obtained from nature, and it is contaminated or it is adulterated in large extent. So we have to assess the raw material. We have to assess the manpower, whether the manpower is performing the desired process, whatever is prescribed in the protocol. Then machine and stability. So these are different parameters which in collaboration decides the fate of the formulation. Now, after coming to the extraction or after coming to the discussing the standardization protocol, we come to extraction. What is extraction? Extraction is the selection of the essential ingredient from the raw material. It is the medicinally active portion which is present in the plant material, and we have to isolate that. And this contains the valuable part. So instead of the entire whole plant or whole plant part, we are interested to isolate the desired phytoconstituents. That a particular process or separation process is known as extraction. It is like it is very tedious process and it requires great skills. If I explain you in the, or if I give the example, there are almost 1500 How is it visible? Huh? Okay, continue. No? Okay, okay. Okay, so we are discussing about extraction. Now, standardized extraction is the where we want to attain the therapeutically or desired portion from the entire bulk of the raw material. And here we use the material which is known as menstruum, which is a solvent or the solvating agent which dissolves the desired phytoconstituent from the sample. Now, your extract may be in the form of tinctures, maybe as a fluid extract or dried extract, depending on the uh, process, and which can be further incorporated into the final doses. So, or maybe solvent, ethanol may be solvent, methanol may be solvent, chloroform, ether, or acetone. So these are polar solvents and non-polar solvents, and with decides because plant contains large number of phytoconstituents. If we are interested to extract the aqueous or water soluble fractions like anthocyanins, starch, tannins, saponins, or terpenoids, or polypeptides, lactins, so they are soluble in waters. For that, we should use these are polar in nature. So for that, we need a polar solvent. Or if we are interested to get some phytoconstituents like fatty acids or components, so they are soluble in ether or non-polar solvents. Similarly, methanol is the one which is semi-polar in nature, and it dissolves both the polar compounds and non-polar compounds as well. And selection of the solvent, its concentration, the time of extraction, these are very important parameters, and which is to be, they are to be considered after a sufficient study or practice. So these are the steps which are involved in herbal drug extraction, like collection of the raw material, collecting the desired plant part from the raw material, drying of the raw material. After drying, the pulverization or powdering or size reduction is there. Then this particular powder or the porous powder is used for either maceration or percolation or different extraction protocols like digestion or decoction. Then filtration or separation, then concentration, and then concentrated extract can be tested for animals or animal studies or for formulations. So these are the steps which are involved, and some of the techniques which are there for plant extracts like maceration, infusion, percolation, digestion, decoction, hot extraction or succinate apparatus or succinate extraction, and aqueous alcoholic extraction by fermentation like Asal and Arista, the Ayurvedic formulations, counter-current extraction, microwave-assisted extraction, extraction performed using ultrasonic or sonication, then supercritical fluid extraction, phytonic extraction. So these are various types of extraction which are there for various products. Not same process is used for all products. It varies from product to product, and the desired phytoconstituent or group of phytoconstituent which we are interested in. Like in case of maceration, we just keep the powdered material in the solvent for a longer period of time, maybe 48 hours or 72 hours. In case of infusion, we just give cold water or boiling water. In case of digestion, we have to boil it. In case of decoction, again, we either boil it or we boil it. And then in case of percolation, we use percolator. So these are the things which are there. These are the techniques which can be used for extraction purpose. These are different, again, percolation extraction protocols like filter machine, then Neapolitan machine, then Mocha machine. So these are different machines which are used for extraction. So this is the demonstration of how succinate apparatus works. This is very common in herbal drug laboratories or pharmacognosy laboratories in pharmacy colleges or in a herbal drug research, whereby we just keep the raw material in this particular thimble. We put the solvent here. We heat the solvent with the help of a heating mantle. The solvent evaporates, go from here, and then come in condensate, condense, and the condensate is again refilled in the thimble, and the extracted material which is soluble in this particular solvent comes from the siphon tube and it is collected here. Later on, this round bottom the extract from the round bottom flask is collected and concentrated with the help of rotary evaporators or other techniques. Now, other tools like sonication is there, counter current extractions there, or supercritical fluid extractions are there, because the phytoconstituents which are present in the plant, they are generally present in the vacuole of the plant, and the main concept of extraction is that we should take the solvent which penetrates the cell wall, which enters to the inside the cell wall, dissolves the material, take out the material, and we can recover the solvent is recovered, so we can get the extract. Now again, this for aromatic compounds, different methods are, or products are like essential oils are there, concentrates are there, absolutes or pomades are there, or resinoids are there, which are traditional methods of extraction like water distillation or steam distillation, coagulation, maceration, and infiltrage. So these are the different methods whereby the extraction of phytoconstituents can be carried out. Now, this is the method whereby we just put the plant material or delicate plant material which contains essential oils like flower petals on the fatty layers, and this particular process is known as infiltrage. We take the layers of fatty material and or wax, we spread the petals, and again a second layer of fatty material, and we keep it for imbibing of the phytoconstituent inside the wax, and later on the wax is melted or heated. So from the molten wax, the flower petals are separated and the essential oil can be distilled off. Now, the discovery from plants, again, for this, there are various steps like collection of raw material, then botanical identification, stabilization, like drying, freeze drying, or lyophilization of, then grinding and powdering, extraction protocols, then qualitative analysis by chromatographic techniques, isolation of phytoconstituents, purification, and fractionation. And this discovery protocol, which is early discovery, then basic research, which is carried out in research laboratories or uh, in academic institutions, up to preclinical studies. The entire study is carried out in laboratories or in academic institutions. The clinical development and post-clinical development, or its FDA approval and clinical trial 1, phase 1, phase 2, phase 3 trials. These are carried out in industry or in large scale. Now we see one by one these steps, like what is the source identification. So natural products is the source of these products, which we can take from our ancient knowledge or from natural or from ethnobotanical approach. Then what is the advantage of ethnobotanical approach? They are pre-selection of potentially active resources are known. The promising safety profile is known. Why? Because like turmeric. So we have been using turmeric for generations. So we know that, okay, turmeric or the phytoconstituents present in turmeric are safe for human being. Similarly, they are cost effective and comparatively they are fast. So if we go for ethnobotanical approach or the traditional approach, then we can get the material. Then in case of phytopharmacy, we take the raw material, its complete extraction, and these are the different extracts like dried extract, liquid extract, tinctures, or encapsulatable mass. So from where we can develop the soft capsules or liquid drops or ointments or tablet or hard gelatin capsules. After this, we have to go for the monograph preparation in pharmacopoeia. We can check for what are the identification tests which are mentioned in the monographs, like for chemical substances. So we go for chemical characterization of the phytoconstituents which we are targeting. What is the appearance and solubility of these particular products? Then what are the identification, whether we have to go for microscopic evaluations or physicochemical tests of the products. Then we go for qualitative analysis and quantitative analysis, and impurity profiles. Impurity profiles may be in the sort of pesticides or microbial load which is present in the sample. And similarly, parallel approach can be used here for the extraction of or druggability evaluation of the phytoconstituents from plants. Like if we take the plant, then for primary assays, you can take methanolic extract or 100% methanolic extract, 50% methanolic extract, or 100% aqueous extract. So this is the stage 1 extraction. After this, say methanolic extract, we 50% methanolic extract we take. So we can further fractionate the extract using hexane fraction, chloroform, butanol, or aqueous fraction. And out of these, we can select the most purified, most active fraction. So we say that it is by acid guided fractionation. And after this fraction, like chloroform fraction. So this secondary assay, we can go for compounds like compound 1, compound 2, compound 3. So we can get the purified compound, and after getting the purified compound which shows the activity on bioactivity guided fractionation, we can go for its structural elucidation. And once its structural elucidation is carried out, then we can go for whether the same compounds can be synthesized in laboratory. Now, in case of preclinical development, we can go by in vitro or in vitro profiling and in vitro toxicological studies like biochemical assays, like tests for enzymatic studies, or in case of cancer cell lines, we can go for cell line studies, or isolated tissues also we can go for with the minimum quantity of isolated fractions. Similarly, we can go for the toxic effect of the fractionated phytoconstituent which we have isolated. Once that is there, then we can go for the working with the cell lines or cell cultures. This is the a new technique which is coming up for last 10, 20 years or 30 years, whereby instead of going on into the animals, or we just go by cell line studies, and this is uh, fast method of analysis. Again, in case of in vivo, after evaluating the drug in in vitro conditions, we go for its preclinical evaluation on animal models like mouse or rat, whereby we check the drug action either by its behavior and reaction, physiology, histopathology, and toxicological like studies like acute toxicity, subchronic toxicity, how much dose of drug particular drug is killing the animal or 50% of the animal. So we calculate that. Then tissue specific toxicity can be evaluated, or tolerability can also be evaluated. Then clinical studies, we can enhance the production or simultaneously we can enhance the production also. This actually is performed in slightly large scale, whereby we can perform the uh, studies. And then next is the phase one trial. In phase one trial, we use 20 to 30 healthy volunteers. Now we are not using the patients for that particular disease for which we have developed the dose or the phytoconstraint, but we are here taking the healthy human volunteers. And what we investigate for the safety and tolerability of the drug or a dose. Then we can check its pharmacokinetics or pharmacodynamics of the drug material. In next step, we on the success of previous study, on the success or basis of success of phase one trials, we go for phase two trials, whereby one day we take 100 to 500 patients. Now we are taking, instead of human healthy volunteers, we are taking patients, and we investigate for safety and tolerability, and then PKPD, then efficiency, doses, and we also design for the dose comparison. Now in phase three trials, three studies, we go up to the number of patients like 100 patients or maybe more than that. And phase four trials is the post-marketing surveillance. We call it as post-marketing testing or post-marketing surveillance. After the drug is launched into the market and it is in the market, then again we have to check whether the drug is showing any toxicity or any adverse effect to the society. And then simultaneously we have to increase the production capacities also, whereby we perform good manufacturing practices and good laboratory practices for the higher quantity production of the phytoconstituent. And simultaneously we have to go from phase one, phase two, phase three trials for the regulatory body also. We have to approach for regulatory body also for the permission and for the getting the required permissions from the FDA department. Next comes the another technique in herbal drug technology is plant metabolomics. In case of plant metabolomics, it is the analysis of the entire metabolome, which can be detectable chemical compounds instead of individual compounds. We have seen in the previous two methods, whereby we use only marker compounds or the individual markers like ascorbic acid in case of Aula, or ursolic acid in case of tulsi, or eugenol in case of clove. These are the phytoconstituents which are the marker compounds of the sample. But apart from that, plants contain hundreds of or thousands of phytoconstituents. And if we see the entire metabolome, these metabolites which may be used available in plants in small quantity may be useful for their therapeutic activity. So for this, the protocol like is applied like herbal medicines. We go for its phyto extracts, then we compare its metabolome by GCMS or LCMS or NMR, and then we perform the multivariate analysis. Or again, these are the statistical tools which are to be applied for the study of different phytoconstituents and their correlation. Now, this is applied for functional genomics, then uh, functional proteomics, and the entire data can be correlated. So, metabolomics, genomics, proteomics, they all come together to give sufficient information about the plants or cells or tissues of the plant or their action. Now, which particular method can be used for the extraction of phytoconstituent? Because here it is important that uh, we say that we need to analyze the entire metabolome, all the metabolites which are present in the plant. So this is very important. So for this, there are various methods which are traditionally used in herbal drug research like HPLC or HPTLC using UV, then GCMS or GCMS, LCMS or MSMS or NMR. So we are concentrating on NMR-based metabolomics, while we'll see in this particular slide. If we prepare sample, so sample preparation in HPTLC or HPLC is slightly easy. They are may not be reproducible. The absolute quantitation in case of HPLC is difficult. The relative quantitation is less. Now, relative quantitation means if you are having the desired marker, then only you can compare the peak of that particular compound in your sample. If marker compound is not there, then it is extremely difficult to check whether the same peak is of your desired phytoconstituents or of other phytoconstituents or of the intermediate. So this relative quantitation is there. Then identity is there in case of HPLC or HPTLC. Then number of compounds by HPLC or HPTLC can be around 30. But and its sensitivity is so. Whereas GCMS, we use GCMS and LCMS for the phytoconstituents which are there, maybe aromatic compounds also. So in this case, sample preparation is not that much problem. Reproducibility is okay. Again, we have to compare in case of GC or LCMS. Now, if we compare GCMS, LCMS or MSMS with that of NMR. So the NMR is, in case of NMR, the preparation, sample preparation is okay. They are reproducible. The absolute quantitation is there because the NMR is the physical property of that particular compound, number of protons present in that chemical moiety. So that is there, which we can evaluate what is the carbon-carbon linking or what is the position of proton. So that can be analyzed in NMR. Now here are the quantitative, if we consider the relative quantitation. Now this is very important. In case of NMR, we need not know, we need not require the desired marker compound. One thing. Another thing is that in case of NMR-based metabolomics, the sample, whatever we are taking, in case of HPLC, HPTLC, or your LCMS, GCMS, or MSMS, that particular sample is destructed. Whereas in case of NMR, the same sample we can use again and again. So because we have seen in our previous discussion, whether in extraction or in primary secondary metabolites, that these secondary metabolites are present in plant in very minute quantity. And if we lose those phytoconstituents in these analytical tools or analytical methods, then they may be, we cannot use those for compounds for formulation purpose or further studies. So this is the limitation of MSMS or LCMS or HPTLC or HPLC, but this is the plus point in case of NMR, because NMR is non-destructive. However, if we consider LCMS or MSMS, the number of compounds are the based on molecular weight or number of fractions. But in case of NMR, number of samples can be analyzed maybe around 20. And as compared to HPTLC or GCMS or LCMS or MSMS, the sensitivity is not that much. Now, this particular slide, this is the photograph of my mentor or my supervisor in Netherlands, Young. So he demonstrated this particular slide. So we want a holistic approach. We want everything in NMR. It is possible in NMR. Whereas the sensitivity of UV or IR is very faint, vague or very faint. Like IR cannot disclose only hello, hello. Okay, okay, okay. So we are discussing about the precision of NMR. Yes, is it audible? Pardon? Okay, 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 okay. No problem. Hmm. Okay, okay. So this particular slide, this is the NMR. So this is a holistic approach. Whereas in case of mass, it is very precise. Or so in case of herbal drugs, we want the holistic approach or the analysis of different phytoconstituents. Now, what are the advantages of NMR-based metabolomics? It means simultaneously wide range of chemical classes of compounds can be analyzed. Like in case of other method, we cannot analyze all the phytoconstituents like terpenoids or glycosides in one run. For that, different solvents are used. But in case of NMR, same sample can be used for all the analysis of the chemical class of compounds. This is a powerful structural elucidation tool. Whenever we discuss for the structural elucidation after the purification of the phytoconstituent, it is a very important tool as it depicts the position of proton with respect to proton and with respect to carbon, whether the double bond is there, triple bond is there, or what is the positioning of protons in the structure. Then this I already discussed, the non-destructive method. It is high throughput because no sample preparation is required. Only once that it is filled in the tube, and same sample can be used again and again. It measures the molecular interaction and concentration, and it is easy to digitalize the data for statistical tools. Now, this PC and PLSDA, these are the principal component analysis or the statistical tools which are used to analyze the huge data. In case of metabolomics, the large or huge data is there, which is to be analyzed with the help of statistical tools, and it is independent of instrument. Suppose the same sample, if I run in India, in Poona or in China, Chennai or anywhere for NMR, because NMR is the physical property of that particular sample, it remains same irrespective of the instrument or irrespective of the place. So now these are some of the examples of proton NMR of ephedrine and their analogs, where ephedrine, methylephedrine, and pseudoephedrine are there, and they can be, and there are various species of ephedra, like Ephedra sinensis and other species, and they contain similar compounds. Similar peaks are obtained in chloroform fraction, but there are other compounds which are there in Synesia, intermedia, or the other species of ephedra. So this type of adulterants can be used using NMR-based multivariate analysis or NMR-based plant metabolomics to analyze all the metabolites which are present and which can be used for differentiating the species. Now these here, if we perform the principal component analysis of the phytoconstituents which are there, we identify in NMR, they can be used like this. So all the compounds which are there in Ephedra vulgaris is collected here. All the phytoconstituents which are there in this Ephedra, this Tachia, is again can be shown here. Ephedra sinensia can be again shown here. So This type of differentiation is possible only in NMR or NMR-based metabolomics. Again, here, what are the similarity or dissimilarity in the Taiwanese ephedra or the herbs, ephedra herbs which are there in the Taiwan market. Now, what happens in case of NMR? What we do, I'll just give you a brief, like whatever peaks are there or bins are there. So we just differentiate that or integrate that signal into the 0.04 ppm. So what happens? The small fractions are there, and the same peak is divided into a huge data. And now that particular data can be analyzed by multivariate data analysis. I'm not going into the details of this because being biological students, I have just only used the softwares. But what is the mathematical background or the statistical tools they are using? That I am not aware of the basic techniques or theoretical aspects. Now, this is the particular study which we perform in Poona, and in the samples are taken to Netherlands for NMR-based metabolomics of Vidania somnifera. So we have collected Vidania somnifera from 4 regions in Maharashtra, in Poona, like Vadgaon, then Dhanukar roots, Srigarh roots, Barje, and Karvenagar. If we see these 5 samples, these 5 samples are hardly around 300 or 3 kilometers or 5 kilometers away from each other. Still, these samples shows variation in their sample, like BR. So this is BR, this is the Vadgaon roots which are showing the phytoconstituents which are present in that. Same Vidani sample which is collected from Poona itself, or say CR. So Dhanukar. So this is the phytoconstituents which are collected from Dhanukar colony, or DR is the Srigarh root. So this much variation in phytoconstituent is can be evaluated using NMR-based metabolomics. These are important because when we go for the action of phytoconstituents which are there in the plant, so and we have seen that the on not only the secondary metabolites but primary metabolites also play an important role in the activity of plant. So for that, this type of study is very important. So same DR means Srigarh road, then Karve road, FR. So this is the enlarged version of that particular pictorial graph. Now these are the proton based NMR profile of Vidania somnifera extract in different solvents. So some peaks are there, and these on the basis of different solvents, the peaks are varying. So the application nowadays, the herbal drug industry, especially when we say that China is leading herbal manufacturer, herbal drug manufacturer, and it occupies almost 70% of the herbal drug market in international market, then they are using nowadays metabolomics or metabolomics based quality evaluation of phytoconstituents or the herbal products in their country. Now, last one is the medicinal plant biotechnology. We see that there are certain limitations of conventional process of procuring plants. Means we have to get and we have to take the desired harvesting plant part, then extraction and purification of the desired phytoconstituent. Medicinal plants are not readily available. There may be geographical or governmental restriction. Then there are issues like they require huge area of land which otherwise can be used for the cultivation of primary food crop. The entire cultivation process is labor intensive and time consuming. The entire the quality may be affected by unforeseen environmental conditions like drought or flood or earthquakes like that, and demand and supplies are difficult to manage. So can we use any alternative methods whereby we can utilize the phytoconstituent or the desired medicinal product from plants? So yes, for that the concept of plant tissue culture is there, which defines that it is an in vitro cultivation of plant cells or tissues on an artificial nutrient media under controlled aseptic conditions. So what happens in plant tissue culture? We take the plant material, live plant material, because we want to use the living entity of the plant material. We perform the entire studies in a laboratory. We provide all the nutrients which are available in soil, and it contains all nitrogen, phosphorus, sulfur. Then we also provide growth hormones and major nutrients, minor nutrients, and we perform the entire experiment under uh, controlled aseptic environmental conditions. So this is just a protocol of Morazic and Scoop's media, whereby it contains major nutrients, minor nutrients, as I told you, the inorganic salts which are present in soil, which supplies nitrogen, phosphorus, sulfur in salt form, and other elements in salt form. Then FEDTA, then vitamins and amino acids, growth hormones, sucrose, because in nature, plants are synthesizing their own carbohydrates through photosynthesis. Whereas in case of tissue culture system, we have to give them ready-made food in the form of sucrose or other fructose. Then we can add agar-agar for solidifying agent. The entire media is prepared in double distilled water and pH can be adjusted. Now, these are the various steps which are involved in medicinal plant biotechnology or tissue culture, like selection of particular media and selection of plant, sterilization of both media and explant as well. Then aseptic transfer of the explant into the medium. Then incubation of at controlled environmental conditions. This is very important, whereby we provide the photo period. Next slide will show that photo period, then temperature and humidity, and it respond in the form of initiation of cellular mass or shoot formation, depending on the which type of phytohormones we have or the growth hormones we have put in the medium. And then we just go for high yielding, fast growing cell lines. And these high yielding, fast growing cell lines can be utilized by different biotechnical strategies to enhance the or the phytopharmaceuticals. Now, these are the culture conditions like photo period, then temperature. We have to maintain the temperature of 25 plus minus 2 degrees centigrade, and humidity 60 to 80 degree, 80%. And photo period we maintain like day and night. So 18 hours, 16 hours a day and 8 hours night we can maintain. Now these are the pictorial representation like initiation of callus, then callus initiation, then from callus to suspension culture. Then this suspension culture can be utilized for different biotechnical strategies like optimization of culture conditions, precursor feeding, micropropagation, elicitation, hairy root culture, bioreactor technology, immobilization, biotransformation, genetic engineering, metabolomics, edible vaccine, so many. So out of this, I'll just give you a brief description about hairy root culture. Hairy root culture is a technique whereby we just transfer the agrobacterium or the oxygen synthesizing gene which is present in agrobacterium, and agrobacterium can be isolated from soil. So agrobacterium rhizogenous or agrobacterium tumefaciens, and this particular bacteria, with the help of this particular bacteria, we infect the plant material. So the bacteria go inside the plant and it transforms the genetically transforms or add the oxygen synthesizing gene into the plant particular genome, and then it starts initiating the shoots. So these are roots. So these are hairy roots, and the difference between hairy roots and normal roots is that these hairy roots develop laterally, and they are genetically very stable, and these can be utilized for the production of secondary metabolites. Once this particular raw material or the hairy roots are obtained, then we can go for different techniques like we have seen here, like precursor feeding. Precursor are the phytoconstituents which are the building blocks of that particular active phytoconstituents. Like I'll give the example of alkaloids. So if we take tropane alkaloids or tryptophan. So if we add tryptophan, then in case of tryptophan is the precursor for indole alkaloids. So if we add a good amount of tryptophan into the medium, it is quite possible that the plant cells which are live cells which convert the raw material of tryptophan into the desired phytoconstituents. So that is the concept of precursor feeding. We can also go for endangered medicinal uh, micropropagation of endangered medicinal plants, whereby we can cultivate those because of now continuous deforestation. So many plants are endangered. So we can preserve those plants by micropropagation techniques. Elicitation is again a concept whereby we just instigate or induce the plant or ask the plant cells to produce desired phytoconstituents. Suppose as we have discussed in the case of Artemisia annua, so if, that produces anti-malarial compound artemisinin. So if we take the Artemisia annua cells and we infect those plant cells with Plasmodium falciparum or Plasmodium vivax, which is the cause of malaria. So it is quite possible that now the plant cells which are growing inside the culture vessels or inside the test tubes or inside the conical flasks, they will synthesize the desired anti-malarial compound from the plant. So these particular protocols are now nowadays used for the production of secondary metabolites. Now, once this liquid medium is or the hairy roots are obtained, then these hairy roots can be used for increased production of secondary metabolites, elicitation or permeabilization, phytoremediation. So many techniques can be applied, and from laboratory scale to pilot scale to commercial scale, scale up is possible in case of hairy root cultures. Now, this is the integrated approach for herbal drug research, like metabolomic profiling, which we have seen in plant metabolomics. So we can go for metabolite fingerprinting, metabolite synergism, metabolic analysis, or targeted compound analysis. And again, omic studies, proteomics or metabolomics or genomics. Then we can apply the systems biological approach, the metabolomic studies and intensive studies of traditional medicine. So they give the hint or the lead identification through metabolomic studies. Then we can also go by evidence-based medicine, and then we can also go for the regulatory aspects of the drugs which are already there in our traditional systems of medicine, but not described in or they are not marketed properly. So we can go for their quality control, we can go for their safety profile, we can generate sufficient information about their efficacy and study, and we can generate the data which is required by modern scientists or modern scientific people for the development of these. If you see the recent development of coronil drug, which is developed by Patanjali and Baba Ramdev for corona. So they claim that their formulation or their kit is very useful for coronavirus. But what about their evidence? What are the scientific explanations of this? So these can be carried out simultaneously. Then conservations of traditional medicines, then ethnopharmacology or biocultural and diversities, ethnopharmacological perspective studies, international cooperation, then global trade. So these are various interdisciplinary approach which can be applied in herbal drug technology or herbal drug research, and which can lead to new chemical entities, new phyto drugs, new formulations, personalized medicines for the well-being of human beings. Now here I'll give you the small example of our effort which we use for the preparation of American herbal pharmacopoeia. They have prepared a pharmacopoeia on Bellerica or Terminalia Bellerica Baheda, and this pharmacopoeia is the manuscript or monograph which contains these parameters like nomenclature, botanical family, pharmaceutical name of the plant, pharmacopoeial definition, then common name of the plant. So this is there. Then historical development or historical information about the plant material, identification. What are the botanical identification, macroscopic, microscopic identification. Then commercial sources. What are the source of procuring that particular raw material? whether it is cultivated or propagated or collected from wild, what are the diseases which are there in the trees or part of terminal Bellerica, then handling and processing, drying, storage, substitution and adulterants, preparation, qualitative considerations. Then not only that, then they need the detailed constituents like what are the phytochemical constituents present in the drug, like terpenoids, tannins, polyphenols, lignans, various categories. What are the analytical tools which are there, which can be used for analysis of Bellerica or analysis of terminal Bellerica or the phytochemicals present in that, like HPLC, HPTLC, UHPLC, and then therapeutics as well, like pharmacokinetics, clinical efficacy, and pharmacodynamics, conclusion of the entire. So these are all the details which are there for the preparation monograph. So this particular information, once is there for the entire material, then we can assure that okay, whatever raw material is there, if we are having one particular sample, so that particular sample can be compared with that of the standard monograph. Now there is a great scarcity of the monographs of herbal drugs, or in India or in society as a whole, and because that is the main hurdle for further research in herbal drugs or herbal drug development. Now this is one of my book which is there for medicinal plant biotechnology, published by Career Publication, and with this brief overview of different fields under herbal drug technology, and these are all different intermediates. Now generally I used to explain herbal drugs or herbal medicines with that of modern medicines in one way, like the herbal drugs or the modern medicines or synthetic medicines are like they are multiple choice questions, MCQs, with negative marking. If your drug or if your entity go to the desired target, then it hit and it will give the positive results. If not, then it will give side effects. Whereas in case of herbal drugs, they are like essay type questions or theoretical answers. They may not cure the entire disease, but they cure the disease from root, and they'll show some results. May not be 100%, but 70%, 80%, 90%, depending on the how it is taken or prepared by the patient. And not only that, one surety is there that in case of herbal drugs, there are very less or no side effects, whereas synthetic medicines, they contain side effects. Now this is the time. This is one slide which I love very much. This is the mural which I obtained from Modern College of Pharmacy in Poona itself, and they have just depicted this one. This is very important. Like we are using traditional knowledge with modern equipments, like modern techniques, maybe HTA, high throughput screening, or principal component analysis, or statistical tools. Now these can be utilized for earning the knowledge. In Corona scenario, in our second slide, this is the technique which we are using nowadays. We are using the net, we are using online teaching learning protocols for learning the basic concepts. And he's Dr. Robert Warput, who visited our lab in Poona College of Pharmacy. And I will, with one prayer, Om Purnamadaha Purnamidam Purnat Purnamidachyate Purnasya Purnamadaya Purnamevavashishyate Om Shanti Shanti Shanti. With this prayer, I conclude my presentation on herbal drug technology. Now I welcome the organizers to ask the questions which are there from the audience. Yeah.
Yeah. So thank you, sir, for a wonderful session. And I would like to have some of the questions from the audience. So the first question is there any references in old explanations like some Samhitas about COVID-like diseases and their cures?
Not exactly on COVID-like diseases, but yes, on infections, there are a number of references are there in our Samhitas, and they do not say that on infection. What they do, they suggest the immune boosters or immunomodulatory agents. If you take these like ashwagandha, shatavari, tulsi, amla, if you or say chawanprash as a whole, if you take these type of materials, then your body will be free from infection. So indirectly, they suggest that yes, these type of preparations are there described in our ancient text, which supports the fight against infections. Not on corona, any sort of infections, because corona is the recent development. But again, it is a sort of infection, and it affects the immune system of the individual. So if your immune system is very strong, then you can fight against it, or vice versa. So yes, the description is there for immunomodulator, and it is also known as rasayana drug, which is used for the vigor and vitality of the human being.
Okay, sir. And another question is like, are you performing any organocultures in your lab? Organocultures.
In our lab, we have cultivated plant cells and tissues in artificial nutrient media, but organic culture, organic farming, we are not, because we are pharmacy college. So we are not having farm or agriculture practices where we can grow the plants or the crop in using cow urine or cow dung or manures or complete organic farming. We are not performing. Yes,
No,
we are
not
performing.
organic, sir. Organic, like root culture, shoot culture, like that.
Or yeah. That figure I have shown you. This is from our lab only. This one, organ.
So in that, like for root culture, what medium
This
you are using, sir?
For root medium, we have to use this one. This is in our lab only. All these slides are my original work. For root cultures, we give good amount of oxygens in the medium, and the root initiates from the oxygen-rich medium, and then we can get developed. In fact, we have also seen not only hairy root cultures, but unorganized roots are also developed in our laboratory, where we have just taken the root and given the rich medium, which contains good amount of oxygens, and then we have developed the untransformed roots from there. Yes, that we have done.
Okay, sir.
Now, apart from that, we have also cultivated one for micropropagation of Gymnema sylvester or Gurban, and we have developed around 400 plants with 95% survival rate, and then we have transferred those plants, propagated plants into the fields. So that is also done.
Okay, sir. And another question is like, what are the chances of remains of solvents after extraction process? If we use any kind of alcoholic solvent, can we use the extract for antimicrobial activity immediately after extraction process?
No, because in case of alcoholic solvents, the solvent residues, there are chances in the extract that solvent residue will remain. So suppose you are taking methanolic extract or alcoholic extract, and if you are performing antimicrobial activity, so you cannot judge that whether the activity or because of the presence of solvent or because of your extract. In that case, your extract should be completely dried. So initially, what we suggest that your extract should be concentrated using rotary evaporators, and then it should be dried. And if it is completely dried, then the chances of residual solvent in the extract is minimum. So that is there, yes. But suppose if your extract is semi-solid, or if your extract is sticky in nature and not completely dried, and that is the common problem with herbal drug researchers. In that case, yes, it is quite possible that the residual solvent may be there. So precautions are to be taken care of.
Okay, sir. And another is like, is there any ratio for extraction? Like you should use this much ratio only, like the indicated, or like it depends on the compounds present in the plant material?
Both ways. Actually, it depends on the phytoconstituents also, which type of phytoconstituents are there. And if not, then we can take around, say, for 100 grams of raw material or plant material, we can take around 1 liter or 700 ml to 1 liter of the solvent. Again, it depends because suppose 100 grams of leaf powder is there, so it is very bulky. And if same 100 grams of root is there, so it is compact. So these are the difference. If
Okay.
your drug is bulky, we have to take more solvent. If drug is hard or compact, then you need
We can
slightly
take
less
less
solvent.
thing. Okay. Yeah. So another is like how
Yeah.
to preserve the herbal extract. Like
This is very important. Actually, herbal extract preservation is once your extract is completely dried or concentrated one, then you can preserve it in a deep freezer, in a closed vessel in deep freezer at minus 20 degrees centigrade or minus 80. Uh, in the Netherlands, we used to preserve those extract in liquid nitrogen also. Here in our college, we preserve in deep freezers.
okay, sir. And another question is like, are you using any endophyte along with plant tissue culture to enhance the medicinal value?
In fact, that is the area of research I am interested to do, but I could not do. I have studied some basic concepts of endophytic fungi or endophytic cultures, but
Okay.
I could not start that work. Actually, that work is to be started. Yes.
Okay, sir. So as of now, this much only we have received, sir. So with this, I think if participant, if you have any questions, you can post it. So, give me 2 minutes, sir. I'll just check if there are any new questions. Okay. Yeah. So this much only questions we have received. So I think I can conclude with this thing. So hello everyone. I hope everyone had gathered great knowledge through the lecture of Dr. Ajay G. Namdev sir, and it is time for conclusion. I am here to propose vote of thanks. First of all, I would like to thank our special guest Dr. Ajay G. Namdev for accepting our invitation and delivering an insightful presentation. The way he handled this lecture was really inspirational, and he gave as much as possible information to all the participants. He enlightened us with full of information regarding the herbal drug technology. After listening today's talk, I hope many participants will look plants as a resource for several medicines, and researcher will focus on their research on developing drugs from the plant resources. And also he enlightened the history of drug technology and the contribution of Indian medicinal plants, like medicinal plants and system, and the various fact and techniques involved in drug technology. He has entirely like he given us a, like, step by step he explained everything in each and everything in detail. So the session was completely full and full of information. For this I would like to thank Dr. Ajay Namdev sir. And also the entire session we had some of the technical glitches. He managed all the things, and immediately, like within the second, he rejoined and he connected with us, and he presented that thing. So for that also I personally thank him. And all the session was entirely informative and helpful to all the participants. Hence, on behalf of e-teaching Tamil and Annakkili Research Institute Chennai, I heartily express my sincere thanks once again to Dr. Ajay G. Namdev sir. We always look forward to your able guidance, sir. Next, I wish to thank the wonderful participant who gave full support during entire session, even though we missed some of the minutes for the due to technical problems. Even though that happened, till they stayed and listened the entire session. So I wish to thank all the participants who supported us, and here I would like to thank Chief Scientist of Annakkili Research Institute, Professor B.B. Chaugli sir, for suggesting Dr. Ajay G. Namdev sir for this presentation. So, and also I would like to thank Board of Directors, Scientists and other staff members of Annakkili Research Institute for continuously supporting me in organizing consecutive webinar sessions. I request all participants to visit AARI website for more details and webinar things. And we have posted feedback link in the chat box. You can fill it to get the certificate. The PIN number was displayed on the video. So I hope everyone got the both thing. So you can get the certificate through that. Thank you all for the support.
I also wish to thank Dr. Chaugli sir for introducing me to you and available this webinar. Thank you very much. Thank you, everyone.
Thank you, sir.